Okay, so this is the second part of the influenza lecture. So we need to talk about the genome structure and the structure of the virus. So first up, if we look at the genome structure, remember that it's an RNA virus, it's negative sense, and it is segmented. Those are the key points. So we've got our negative single-stranded RNA in 8 segments. Now this right here is kind of an outline of the genome. We've got our segments here, and of note, all of them are going to encode for one messenger RNA and one gene, except M and NS, non-structural protein. These ones are spliced. That means that they can encode for multiple genes. So M is going to be spliced into M1 and M2. Those are the two matrix proteins, and M2 is the ion channel, and NS encodes for non-structural protein 1 and non-structural protein 2. Now, all of these genes are going to be found, or I should say all of these gene products are going to be found in the virion except for the non-structural proteins. The non-structural proteins you'll only find during an active infection. So this is what the particle looks like. So the particle, you'll notice, has an envelope. It's very important here. And you'll notice that it's drawn as a circle, so this would be how it's found in tissue culture. In the envelope, you have the spike proteins, a hemagglutinin, also abbreviated with just an H, and neuraminidase, which is also sometimes just abbreviated with an N. So these are our spike proteins. Remember, hemagglutinin is essential for entry, neuraminidase is essential for exit. Then we've got M1, and M1 is the most abundant protein, and this kind of forms that underlayer right underneath of the envelope. M2 is the ion channel, and this is what's going to be responsible for encoding. And then we've got our gene segments inside, and on for each gene segment, it's going to be coded in the nucleoprotein. The nucleoprotein protects the genome, the RNA genome, helps protect it from the host innate immune system. And then we've got the polymerase structure. So the polymerase is formed of these 3 genes, or these subunits, PA, PB1, PB2. So there's a really good table in your book, which is table 9.4. And this goes through the functions of all of the genes, which you need to know. So briefly, we'll talk about them here. So these gene products here, these are all involved in the polymerase. They're involved in cap snatching, and they're involved in making more RNAs, making messenger RNAs and making more genomes. You know that hemagglutinin is going to be responsible for entry. You know neuraminidase is going to be responsible for exit. Oops. That's why there's an NP and an NA. I just made a mistake there. So NP is our nucleoprotein, and this is going to coat the RNA and protect it. Coat the viral RNA, as shown here. And then Na is going to be responsible for exit. Now, neuraminidase cleaves the sialic acid residues that allows the virus to leave the cell. But I think it also might be important that while the virus is navigating your respiratory tract, neuraminidase might be able to cleave the sugar mucin to help get through the mucus and actually get to your epithelial cells, which is kind of cool. So, matrix spliced. That means it's spliced just like your RNAs are spliced, so it's got to be in the nucleus. And so again, M1 is our most abundant, and that forms the kind of shell, the protein shell that's right under the envelope. And M2 is our ion channel. Okay, so our non-structural proteins, this is going to only be during replication, so it's not, they're not present in the particle. So they're only during active replication. And these do some very important things for the cell, for the virus. So N2 is going to be responsible for nuclear export. So N2 helps get the messenger RNAs out of the nucleus and into the cell. And then the other non-structural protein, NS1, this one can actually do a couple things. So I'm going to bring this down here. So non-structural protein 1 is important for regulating transcription. It's important for regulating the splicing. It also sequesters RNA in the nucleus. This is going to trap cellular RNAs so they can be, the caps can be snatched, and it's going to bind up viral RNAs until they're ready to be exported. By also protecting the RNAs, the viral RNAs, and the double-stranded RNA intermediates, this is going to prevent the antiviral state from occurring. This is going to antagonize PKR. So remember the antiviral state. Remember the importance of PKR and interferon. So non-structural proteins, NS1 and NS2, are important for regulating transcription and making sure only the mature viral messenger RNAs leave the nucleus at the right time, trap the cellular RNAs in the nucleus so they can be cut apart and the caps can be snatched, and making sure that transcription runs smoothly for the virus. So these are important proteins that you're only going to see during an active replication. They're not in the particles. So again, here's a handy-dandy chart. The influenza replication cycle is going to start with hemagglutinin binding to sialic acid. Then the virus enters the cell in an endosome. It enters through endocytosis. As the pH becomes acidified, it triggers membrane fusion. It triggers fusion between the viral envelope and the membrane of the endosome. Now, this is something that's really hard to show in three-dimensional space. You'll notice what's shown here in the picture. This is from your textbook. You've got the hemagglutinin protein, and it's trying to show you the hemagglutinin protein is kind of like a hairpin, and when the pH acidifies, it extends out. The hairpin unfolds, and then it folds back and it brings the two membranes together. So this is shown, this is a picture I took from a paper. You've got the hemagglutinin here, or the spike protein here. At the low pH, it pops out of its hairpin. It inserts the fusion peptide into the endosomal membrane, and then it clamps back, and it causes fusion of the viral envelope and the endosomal membrane. Okay, so I'm going to show you a snippet from a video on YouTube demonstrating viral entry and membrane fusion.
Everything begins when the virus enters our airways.
To sialic acid. We'll touch base on this a little bit later in the lecture, but in order for that hemagglutinin hairpin to pop open, it needs to be cleaved, and that can happen either in the Golgi before the virus leaves the cell, in which case it's a lot more infectious, or it happens now and some host enzymes will cleave the hemagglutinin. So that, I think you can see the fusion between the two envelopes in the video a little bit more clear than you can in a two-dimensional picture. Okay, so in the video you can see how the membranes fuse. And in the video it mentioned that you had some cellular enzymes that cleaved the hemagglutinin. So what you've got here is the hemagglutinin, and before it's cleaved we refer to it as H0, and after it's cleaved you got H1 and H2. Now if the there's an error, so it should be inside of the cell in order for it to be infectious or before entry. So one of the maturation steps that can happen during influenza infection is in the Golgi apparatus. This can get cleaved, and then it's primed and ready to go to infect the next cell right away. If it's not cleaved in the Golgi, then it still can get released, but it's not fully infectious, and so you need cellular enzymes outside of the cell, and they can either be free-floating or they can be attached to the surface of the cell that are going to cleave right here at the cleavage site. in order for the virus to be infectious. If this doesn't get cleaved and it gets internalized, then the fusion peptide can't pop open, and it can't cause fusion between the viral envelope and the membrane of the endosome, and then the virus just gets destroyed in the endosome. Now, there are some antibodies that specifically bind this region, and then they prevent it from popping open. So one type of neutralizing antibody is going to bind hemagglutinin right here and prevent it from opening up, so it would just get destroyed in the endosome. Other types of antibodies might bind, say here, and then they would prevent it from attaching to sialic acid. The only neutralizing antibodies are ones that are going to bind hemagglutinin. And so those neutralizing antibodies either bind here and prevent it from attaching to sialic acid, or they bind here and they prevent the fusion protein from popping open. So the next step is uncoating. So we get out of the endosome, and kind of right after that, or concurrently, we've got the M2 protein and the M2 ion channel. This is very important. It's pH dependent and allows some protons to get inside the virus. Not too many, but enough. This weakens the other matrix protein, M1, and this allows the matrix to sort of fall apart at the right time, so then the genomes can get into the cell. Now, I want to point out at this point in time that there are a few drugs, antivirals, that can block the ion channel. All right, so one of them here is amantadine and also rimantadine, and these block the ion channel so the genomes can't uncoat. I want to point out here in your book, there's an error in the book. So in your book, on the fusion and uncoating image, it labels fusion here as like entering the nucleus, which is weird. So that's not fusion. Fusion is this process here, right? The fusion of the viral envelope with the endosomal membrane. And then this arrow here, this is uncoating, right? So this is when the ion channel allows the matrix to open up and allow the viral nucleoproteins, coded genomes, into the cytoplasm, and then they traffic to the nucleus. So once in the nucleus, you've got to get your polymerase, and your polymerase complex is your PA, PB1, and PB2. And these proteins, they form a complex that's going to allow for the transcription of messenger RNA and also synthesis of the viral genome. So they're going to enable the cap snatching, right? They're cut up the cellular RNAs, steal the 5' methyl G cap along with the first few nucleotides of the host messenger RNA. This allows it to be used as a primer for transcription. All right, as I mentioned before, NS1, this is important. Non-structural protein 1 is really important for preventing nuclear export of pre-RNAs. So this traps cellular RNAs in the nucleus to facilitate cap snatching. And non-structural protein 1 also binds and sequesters double-stranded RNA intermediates, and this prevents the activation of the antiviral state. The non-structural proteins also enable the alternative splicing, so you get the non-structural proteins 1 and 2, and you get both matrix proteins M1, which forms that kind of shell, and the ion channel that allows it to uncoat. Now, so this is all the information that we've talked about so far in one diagram. Right, so you've got your viral particle, it binds to sialic acid. It comes in through endocytosis. As the pH acidifies, hemagglutinin activates, that hairpin pops out. It allows fusion between the viral membrane, the viral envelope, and the membrane of the endosome, and the ion channel M2 allows a few protons in, and that change in pH weakens the matrix M1 protein and allows for uncoating. The genome, the segmented genome, goes into the nucleus, where the polymerase complex, so we got PA, PB1, PB2, they make new copies of the genome, and they make messenger RNAs. The viral messenger RNAs, some of them undergo splicing, and they use cap snatching. They're exported, and then they're translated on ribosomes. Now, the proteins, the glycoproteins, are going to have to go through the ER, go through the Golgi, and then they're going to collect at the plasma membrane. Everything collects at the plasma membrane, and then when you've got all of your genome segments, you've got your matrix protein to form that shell, then it can bud off, but the new particles get stuck on the cell membrane, so you need neuraminidase to cleave the sialic acid and allow the new virions to escape. Now, we're going to end this segment of the lecture, and then we'll do one short segment to talk about the epidemiology and genetic variation.